Okay, now we are going to move on to chapter 3 and take a little bit of a deeper dive into the individual units of cells. So the big thing to remember with cells within the body, one, there's a lot of different types. There's just a few examples on this slide here. And like it says at the top of the slide here, function dictates form. So based on what we need the cells to do, that's why they look like they do. The way the cell is designed is to help it do its job to the best of its ability. So nerve cells, for example, have long extensions, which will allow them to send signals to other cells. The extensions, like you see here, can be up to 1 meter long, and that's how we send our info. For red blood cells, they're this disc or concave disc, and that allows them to squeeze through some of our blood vessels. You see, muscle cells are elongated. They have fibers that can shorten so that when we contract or shorten our muscle fibers, our muscle cells, we can actually move our body parts. And next, we'll go over white blood cells. So they have the ability to identify foreign invaders, and some of them can even open up and eat those foreign invaders. And then you have gland cells, which have the capability of storing and releasing substances. So as a whole, just remember that function dictates form. So based on what a cell needs to do, that's why it's designed the way it is. All right, now let's get into a little bit more of the pieces that make up the cell and the cell structure itself. So going out to in. So your plasma membrane, that is the outside portion of the shell or the cell. So that is going to make contact with the outside world, and that's what engulfs or keeps everything enclosed. Once you get through the plasma membrane, you hit the cytoplasm. The cytoplasm is kind of all this whitish space in here, and all these other pieces are organelles that sit within the cytoplasm. And then you hit the nucleus. That's kind of the most important part of the cell. That's what contains all the genetic information, that has all the instructions that tell a cell to do what it needs to do. And we'll get into all these pieces more in depth in these next few slides, but this is a good overview picture. All right, so the plasma membrane starting on the outside. So it's very important because it lets in what needs to get in and it keeps out what should not get into the cell. So it's considered selectively permeable, meaning that it only lets in certain materials. So small, non-polar, or uncharged molecules, it can pass through. So the way it does this selectively is having these two layers. So these little blue guys here are known as phospholipids, and you'll see they have heads. So the heads point towards the outside environment and the inside of the cell, and they also have tails. And so the tails point in towards each other. So I'm not sure how many of you would have taken medical terminology yet, but this is a good time to put that to use. So the heads of your plasma membrane are hydrophilic. So hydro meaning water and philic meaning loving. So the heads love water. They don't mind touching water. The tails, on the other hand, are hydrophobic. So once again, hydro meaning water and phobic meaning does not like it. So the heads that make contact with the outside world and the inside of the cell, they're fine with water. No water in here. You also have cholesterol within the cell membrane. That gives it some strength and stiffness. That helps it hold its shape. And you'll also see proteins within the cell membrane. Those are embedded. So they have a few different jobs. In this case, you see they're letting molecules pass. So these may be molecules that would not normally be able to just pass through. So we have to send them through a special tunnel. And then sometimes you'll see one like this, and you see it has its little extension. And that typically is a way for a cell to identify itself to other cells or get an idea of what's going on in the outside environment. And we'll go into that stuff a little more in depth farther down this PowerPoint. Okay, so this is a little bit of a busy slide. This is kind of a summary of what I was just talking about on the previous slide. So like we said, the head likes water, hydrophilic. The tails do not like water, hydrophobic. They form that bilayer, heads to the outside and the inside, tails point towards each other. That's known as the hydrophobic core. So we talked about the cholesterol molecule scattered without. That's going to give some stiffness and strength. That helps it hold its shape. And then our embedded proteins. Like we said, channels, they can be receptors, they can be markers. So a few different jobs for the proteins. All right, back to the nucleus. So this is like the brain of your cell. This is the math computer. This has all the instructions that you need for your cell to know how to do its job. And it's made up of a few different parts as well that we'll go into next. All right, so there's a membrane similar to our plasma membrane, but just surrounding the nucleus, and that's called your nuclear envelope. And similar to the plasma membrane, it has some control over what gets in and out of the nucleus itself. The nuclear pores are embedded within that envelope, and so their perforations are little holes within that membrane. So same thing, they can regulate what's getting out and what's coming in. And so DNA and RNA are your genetic material. And so the DNA is contained within that nucleus. All of our genetic material is contained within that nucleus. And so we need materials to be able to make that stuff, and we need the RNA to get back out so that it can tell everything else what to do. And we'll get into DNA and RNA more in depth because that's very important. So like the cytoplasm is kind of the gel that contains all our organelles. There's nucleoplasm. And so that holds a lot of material and things within the nucleus. One of those important things would be chromatin. So chromatin looks like little threads. They're made up of DNA and protein, and that's what eventually will coil up to form an actual chromosome or a gene. And then you have the nucleolus. That's the very center of the nucleus. And this is where ribosomes are manufactured. Ribosomes are another organelle that are very important in protein synthesis. And that's where those are actually manufactured before they're sent out to the cytoplasm. All right, first review question for this lecture. So which component of the plasma membrane is responsible for maintaining its stiffness and strength? A, phospholipids, B, cholesterol, C, the nuclear envelope, or D, proteins? All right, take a moment, pause your video, think about it, look back if you need to. The answer is B, cholesterol. So phospholipids are that head and tail component that you saw in that picture. We have a lot of them, and they create that double bilayer and give it its hydrophobic core. It's what partly makes it selectively permeable, but it's not necessarily what stiffens it up or gives it strength. The nuclear envelope is that membrane that surrounds the nucleus, so that's actually not in the plasma membrane, even though it has a similar function just for the nucleus. And then proteins we also see within the plasma membrane, but they are used for channels, receptors, and identification. So they're very important, but not for stiffness and strength. So cholesterol gives it stiffness and strength. Okay, going on to organelles. So this is an example of your endoplasmic reticulum. That's the first organelle we'll talk about. So these are separated into two categories. You have a rough endoplasmic reticulum, or ER, or smooth endoplasmic reticulum. So the rough ones have ribosomes right on top, so that gives it the texture. That's why it's considered rough. And then the smooth has no ribosomes on its surface. Okay, so you can think of the endoplasmic reticulum as the highway. So the highways and the tunnels, it has a bunch of canals and sacs that travel from the plasma membrane to the nucleus. So when stuff from the nucleus needs to get to a different part of the cell, it can kind of go through and travel through the rough ER or smooth ER. So like we said, the rough ER is called the rough ER because it has the ribosomes on the surface. And then we have the smooth ER, and that doesn't do as much of the highway function in transporting for ribosomes and some of our genetic stuff. But the smooth ER will synthesize lipids or fats and carbohydrates. So that's its main job. But the rough one is what you'll hear referred to a little more often because it has that role in our genetic material and protein synthesis. Okay, the ribosomes themselves are little tiny dots, granules of protein and RNA. You'll see, obviously, there's ones attached to the rough ER. We just talked about that. And then others are just scattered throughout the cytoplasm. So its job is to make proteins. So basically, it gets the genetic information from our master computer. The nucleus sends out the RNA that has all the instructions written down from its DNA, and it arrives at the ribosome. The ribosome can then interpret that information to create a protein in a process known as protein synthesis. And we'll get into the actual process and steps of protein synthesis down the line. Right now, all you need to remember is that the ribosomes are the site where that happens. Okay, the Golgi apparatus. Here's another organelle. This is your mailman. So you get proteins from the endoplasmic reticulum. They put it in a box, package it up, and then it can export it to other parts of the body. So there's your ER, and this would be a rough ER because it has the ribosomes. Sends it to the Golgi packages it up, and it's going to go out to another cell. All right, I don't need you guys to memorize these steps by any means. I just wanted you to get an idea for exactly how the Golgi apparatus does what it does. All I need you guys to know in terms of quizzes and testing is that the Golgi is what packages and sends out stuff. So in this case, we have a protein molecule is coming from the ER to the Golgi. That's going to then pass through the different layers of the Golgi. It may make modifications to it. It's then going to envelope and package up the protein. And then you see this little bubble. It's going to make a vessel out of its own membrane. And that's called a vesicle. That's then going to travel to the surface. Our plasma membrane will open up and it'll get rid of it. You can also have what they consider a lysosome instead of a vesicle. Lysosomes will kind of destroy or recycle the material rather than releasing it. So that's what that refers to down here. But this is primarily the function here is that export. Okay, so we just talked about lysosomes. So that the vesicle is that little bubble that forms. That's a vesicle. And so they're pinched off from that Golgi, and they have enzymes in them that will break down things. So when we get a protein or something that the cell doesn't need, rather than sending it out to another part of the body, it will just pinch off a lysosome, and that enzyme can break it down. That's a way we can do some recycling. So amino acids, which are our building blocks for proteins, when we need some more of those, we can break down unnecessary proteins back into those building blocks so that we can make new ones. It can also destroy bacteria. So like I said, the garbage disposal. I would think of it more as a recycling machine a lot of times, especially when we talk about it when it refers to proteins. And it's not necessarily getting rid of the protein completely. It's more breaking it down so that we can use those building blocks again. And these are just contained within the cytoplasm. Then you have centrioles. These are also contained within the cytoplasm. And so they sit right outside the nucleus because they have a very important role in cell division. So we'll get into cell division and the steps for that down the line, and we'll talk about centrioles again. Right now, all you need to know, they sit near the nucleus and they are important in cell division. All right, the mitochondria. This is one I would guess a majority of you have heard about. And I'm sure all of you have heard that corny saying that the mitochondria is the powerhouse of the cell. And so this is where all of our energy is made. This is a very important piece of the puzzle. This is how we get our energy to do what our cell needs to do. Like I said, the mitochondria is the cell's powerhouse. And you'll see there's kind of a double membrane, outer and inner. This inner one has folds back and forth, and that forms cristae. So within these spaces in the cristae, there's a process that goes on. So basically, we can make ATP. That's the important part. Mitochondria are going to manufacture and create ATP, which is the energy for our cells. So it makes sense that cells which have to do a lot of work, like a liver cell, are going to have more mitochondria than something that does much less work. So the more energy a cell needs, the more mitochondria that's going to be found because it can create more ATP for the cell to utilize. All right, so cytoskeleton. So this is the framework of a cell, and it's going to determine the cell shape and also give the cell some strength. Sometimes it can allow the cell to move for those that need to. And there's a few different classifications under this cytoskeleton, especially for the extensions. A lot of these are to allow the cell to move. So microvilli are folds of the cell membrane. It creates a lot of surface area. So we can absorb things much better because there's a lot more area to do that. So in the intestines, where we have to absorb all the nutrients from our food, you'll see microvilli. Cilia are a hair-like process. They kind of beat back and forth like they're waving in the wind. They're like waves, and they can help propel a substance. And then you have flagella. That's more like a whip-like motion to move a cell, and you only see those in sperm. So if you've seen what a sperm cell looks like on a microscopic level, it kind of has a head, and then you'll see a tail. That tail is a flagella. That's the proper term for the tail. Okay, second question for this part of the chapter. Which organelle is responsible for preparing, packaging, and exporting proteins? A, the Golgi apparatus, B, the rough endoplasmic reticulum, C, mitochondria, or D, nucleus? Take a second to think about that one. And the correct answer is A, Golgi apparatus. Remember, that's our mailman. He's going to package and export material. So it's getting its material from the rough ER because it's helping to manufacture and deliver proteins to the Golgi. But its job is to get them to the Golgi apparatus. It's not sending them to the outside of the cell. We just talked about mitochondria. That's the powerhouse. That's our power plant. That's where we get all of our energy. And then we talked about the nucleus as well. Its job is to be the master computer. It's got the master copy. That's all the genetic material in the cell. All right, I think we're going to go ahead and wrap up this first part here. And then we will come back for part 2. We'll talk about transport and movement within the cell, and we'll dive a little deeper into protein synthesis and cell division. So take a quick break and then come back for part 2.